All right, it's good to be back. I'm feeling good. My husband's down now, but I'm feeling good because my husband is down. I'm gonna have my Zoom or my office hours by Zoom tomorrow. Just there's some things that he usually does at home on Thursday that now I have to. If you haven't logged into Zoom yet, they're making you do it different now. It's through SSO, and you need a. If you haven't done that yet, you have to. No, it's GVSU-edu. I'm not sure if that works the same as what it does for me, but maybe you log in differently. You should be able to just call my phone number, which is a Zoom phone. I'm not exactly sure how that works yet, but I should get an email but it should ring on my iPad. Anyway, I'm still trying to work that out, but I will be available at that time. I'll just be at home. And I just wanted to reiterate, I sent out an announcement yesterday just so I wouldn't have to spend a bunch of time talking about the exam, which is a week from today. I know it's crazy. It's a little shorter exam. Those are your homework assignments. That homework is due on Wednesday. A number of you haven't started it yet. So I would get started maybe the chapter 3 stuff, wait till we get a little bit more into it after today. Obviously, we won't finish it till Monday, but that is on the horizon and fast coming up. Okay, question.
Will the exam questions be based on the textbook or the notes?
So my exams are based on what we talk about in class. So what notes we take in class and what is already on the PowerPoint. I won't pull anything from the textbook that we haven't talked about. So most of there may be a few homework questions that you had to go searching for in the textbook. I tried to drastically cut down on those type of questions. So yeah, all multiple choice. Good, thanks for asking. All right, I also wanted to put a little plug in for our VMS seminar series this Friday. It's actually a seminar that is very applicable to this class. So if you've never been to one of our seminars, this would be a good one to come to. Dr. Rankema is on faculty here, and she doesn't teach a lot of undergraduate classes because she just got a big grant, but she does work with undergraduate research students. So if that's of interest to you, come check out what she does. It's a lot of immunology research, mice, but I think she'll give it a pretty good talk. So it's over in the basement of MAC. Sometimes that room is kind of hard to find, but so that's again this Friday at noon. Okay. All right, so we have talked about some history and some taxonomy. Hopefully, most of you have listened to that lecture that I gave last week, kind of talking about how we grow bacteria in the lab and some terminology we use, as well as microscopy. So important stuff. Make sure you listen to that. A little overlap with lab if you're in lab. Okay, so we're going to set out now. Chapter 3 is a pretty big chapter. We're going to talk about both prokaryotic and eukaryotic cell structures. So we'll review what those terms mean, prokaryotic versus eukaryotic. Just kind of, we're going to start from the outside and work our way in and look at all the different structures. Hopefully get some familiarity with those. And we're going to spend a lot more time on bacteria than we are on anything else. So, okay. All right, so just a reminder, these terms you should have come in contact with several times in your education before now. But a prokaryotic cell, remember when we talked about that Wose and Fox phylogeny, how he sequenced the ribosomal RNA and he took those prokaryotes and he said, well, they're actually two different domains, the bacteria and the archaea. What makes them, why do we consider them prokaryotic? Basically because they lack a membrane-bound nucleus, and they also don't have any other internal organelles that are bound in a membrane. Like they don't have lysosomes, they don't have endoplasmic reticulum. They do have ribosomes. Ribosomes, even though we call them an organelle, they are not bound in a membrane. We'll talk more about ribosome structure, but there are actually two parts of the ribosome that come together to make protein. So it wouldn't work very well if they were enclosed in a membrane. In terms of size of a typical prokaryotic cell, on the order of about 1 micrometer. Okay, so I talked about those units, micrometer and nanometer, in our last lecture. This is the same thing as 0.001 millimeter. Yeah, okay. So they're pretty small. That's kind of a general sense. Here you can see in the scanning electron micrograph, the units here, one cell, okay, about a micrometer. Contrast that with this paramecium here, where the same, from here to here is 40 micrometers. Okay, so very much smaller, the typical prokaryotic cells, much smaller than the typical eukaryotic cell. So likewise with eukaryotes, kind of all the opposite things. They have a membrane-bound nucleus, they have membrane-bound organelles, Golgi, endoplasmic reticulum. We'll talk about those kind of briefly. The eukaryotes include algae, fungi, of course, plants, animals, protozoa. Okay, we'll talk a little bit about some of the ones that are microscopic. Either we'll start that Monday or probably after the first exam. Okay, the size of a typical eukaryotic cell, typically somewhere between 10 and 100 micrometers. So anywhere from 10 to 100 times larger than the typical prokaryotic cell. Okay. We talked about this a little bit last time. It bears repeating, thinking about different morphologies of prokaryotic cells. So these terms, cockeye, that's a round cell. A rod-shaped cell we typically call a bacillus. That's kind of that didn't come out through very clearly on there. Vibrio, it's a comma-shaped organism. We talked about those. Coccobacillus, somewhere between a cockeye and a bacilli. And then I wanted to tease these bottom two out a little bit more, are helical-shaped organisms. Both are spirilla and are spirochete. Okay, so both of these, they look very similar, just to kind of look at the drawing of them. The spirillum actually are a little bit more rigid. When you see them move, the whole body doesn't flex very well. And they have external flagella. We're going to talk much more about flagella today, but that's just what prokaryotic cells use to move purposefully. And then the spirochetes, their cells a little bit more flexible. And they have an internal flagella. It's actually enclosed in a membrane and runs down their entire length of their body, or cell, I should say. Okay. So here's an example here. Some maybe a little better pictures here of both. And I think both of them. See if I can bring that up. So here on the left, this is the spirilla. Oops. Or spirillum is the singular. This is a scanning electron micrograph. So remember the difference between scanning and transmission. The scanning is more of a 3D view. Sometimes it's kind of difficult to tell if you don't know what you're looking for, but the transmission, remember, is a cross section. If you look closely at that, you can kind of see some brown, of course, this is artificially colored, but that's the flagella on the outside of the cell there. And then here's some spirochetes here on the right-hand side, a longer cell. Any guesses as to the kind of microscopy that is on the right? It could be confocal. We didn't actually talk about that one, but that's a good guess. It's kind of hard to tell from this. By the dark background, a good guess would be dark field microscopy. It could actually be fluorescence without knowing for sure. If this was a video and they were moving around, then you would know that it's dark field. Because for fluorescence microscopy, we have to stain the cells, and when you stain them, you always kill them. Okay, so if it's something that's alive, it's either going to be bright field or dark field, and given the dark background, get much better contrast with the dark field. Let's see, sometimes this doesn't work very smoothly, but let's see if I can bring up. Oh, there it goes. Okay, let's see if I can bring up one of these. It's kind of fun just to see them moving around. So, yeah, so here's some spirochetes. Quite flexible. I think you want to move in that helical way that allows them to kind of move through materials that are maybe a little bit more thick, a little bit more viscous. Okay. So it's kind of fun watching them move around. Let's see. Let's come back. Okay, I think I skipped the other one for now. So, okay, we'll look at some more flagella movement shortly. Okay, so we talked about prokaryotic morphologies of cells. Another thing that we like to look at that helps us tell the difference between different bacteria is how they tend to arrange, the cells tend to arrange themselves. Okay, do they tend to divide in like a single plane or multiple planes, and do the cells tend to stick to each other once they divide? So sometimes you might, for the cockeye, you might see a single cell. If you have a cockeye that only divides in one plane and then doesn't stick to each other very long, you might see some pairs or diplococci. If you have a cockeye that will divide in two different planes, then you could get a group of 4 or a tetrad. There is another term that they don't throw at you here called sarcina. And that's when a cockeye actually develops or divides in 3 different planes. So you picture like an X on the top and then another plane part way through it. It will form groups of 8 or okay, I didn't draw that very good, but that's supposed to be a cube. So it's supposed to be a packet of 8. Okay, so sometimes when you're looking at cells under the microscope, it's kind of hard to tell whether they're tetrad or a sarcina because it's hard to tell the 3D-ness of it. Okay, so anyway, sarcina, a cube-shaped arrangement. Chains, when they typically, again, divide just in one plane and then stay stuck together for a long time. Streptococcus, that's a genus that we're going to talk about quite a bit in this class. They tend to form nice long chains. Staphylococcus, then grape-like clusters. That prefix staphy is Greek for grape-like clusters. So when you're staining cells under the microscope and you see a particular arrangement, that gives you a hint as to what kind of cells that you're dealing with. The bacilli, the rod-shaped cells, don't tend to cluster together in a lot of different arrangements because they will only divide on one plane. They divide kind of up and down. You don't, what is it, like hamburger style rather than hot dog, I think maybe is how you might say it. But they just get bigger and bigger, and then one cell divides into two. So you don't see a lot of different crazy things. It's mostly singles. You can get some chaining of rods. Sometimes you get two cells that kind of will only stay stuck together in like one little spot. And that's sometimes that's referred to as snapping division, but I think we won't bother to write that and define that. But okay. All right. Moving on. So starting on the outside of the cell, what's the first thing that we might encounter for looking at a bacterial cell? Many bacteria cells make some kind of glycocalyx. And I should have written that a little bit bigger because that was the main point of here. So a glycocalyx, what is that? That's some slimy substance that's secreted outside of the cell. I'll change my color, that's kind of hard to see. Okay. And this is really common. Lots of bacteria have this. It can be a really thick glycocalyx, or it can be a much thinner one that's much looser defined. It's going to be made most of the time polysaccharides. There's some variation with that. There's a few organisms that have a little bit more protein composition to their glycocalyx, but most often some kind of sugar surrounding the cell. And like I said, this is very common. Most cells are going to produce this because it has such rewards for the cell. What does it function in? Attachments. That's one function. So if a cell can secrete some kind of glycocalyx, it can stick to other cells and maybe make a biofilm. It can maybe stick to some kind of structure. Like we have lots of bacteria in our mouth that make a glycocalyx so it can adhere to our teeth and not get washed down into our digestive system. There's bacteria that adhere quite nicely to catheters or some kind of implants, okay, because they make a glycocalyx. Also, it helps them avoid phagocytosis by white blood cells. That's a process that we'll talk much more about later in the semester, but any cell that's trying to establish an infection in our body, if it makes a capsule, it's harder for our innate immune cells to clear it. You're going to usually maybe need some help by some antibodies. Okay, so protection from our immune system, as well as some protection from drying out. Okay, a cell, a bacterial cell that's popped out of our lungs onto some surface. If it has a glycocalyx around it, it's going to be viable for longer out in the environment because it's not going to dry out as quickly. Okay, so all important functions, we usually consider that glycocalyx either a capsule, a true capsule, or a slime layer. Just wrote that a little bit bigger there, even though it was written already before. Again, this is a transmission electron micrograph that's been artificially colored just so you can kind of see what's what. But the capsule here is this orange layer outside of the cell. If it's considered a slime layer, that's just a capsule that's just a glycocalyx that just is not as well organized, a little bit less tightly bound to the cell. So you can see it's a little bit, again, not as regular. Okay, if you're in lab, you're gonna get to do a capsule stain, let's see, next week. Yeah, next week. And a capsule stain, that's one where we purposely stain the background, because that capsule, if it's polysaccharide, it doesn't have much of a charge, and our dyes don't stick to it very well. So we usually try to stain the background. It could look something like this. That's not a great capsule stain. The cell walls, they are stained as well as the background, and then anything that's not stained, that's the capsule in that picture. And sorry, somehow all this got kind of cut off somehow when I entered that in. There is a homework question in chapter 3 that asks you basically to label a cell. It's kind of a tricky question, really, and don't get too worked up about it. It's listed just for participation points anyway. So, but that's referring to that diagram, which is in the OpenStax. Okay. This is another example of a capsule stain. This looks a little bit more like the one that we're going to do in lab. You can see this big kind of unstained part around all these cells. That's the capsule, kind of the great big halo around it. So, all right, so very useful for bacteria cells. Eukaryotes can have some kind of capsule or glycocalyx as well. We'll talk about that. So the next thing we come to as we're working our way into the cell is the flagella. Because the flagella typically are not going to be enclosed in a membrane, at least for bacteria. The main function of the flagella is for motility, is to move that organism around towards some attractant. Okay. Lots and lots of bacteria have flagella. It's usually the rod-shaped ones or the vibrios or the helical bacteria. Typically, any bacteria that's just a round cell, we don't see a flagella like that. So there may be some exceptions somewhere, but as far as I know, no cocci have flagella. Mostly, again, the rod-shaped ones. Not all bacteria have them. If they don't have flagella, that just means they're just going to kind of float along with the medium that they're in. Okay, and if they have flagella, they're going to be moving purposefully. So the structure of the flagella, and this is going to be quite different whether we're talking about bacteria or whether we're talking about some kind of protozoa with a eukaryotic flagella. So we'll contrast those later. They're going to be made of a filament, which is composed of the protein flagellin. Kind of makes sense. A filament, a hook, and that's going to be anchored into the cell wall by the basal body. Okay, so you have kind of a hook, a filament, and then there's going to be kind of like some rings where it's anchored into that cell wall. I have another picture of it on the next page, but just kind of an idea. So the basal body, that's where it's anchored into the cell wall. Okay, and like I mentioned before, it's going to be not enclosed in a membrane. There's one exception to that. We'll get to that exception, but generally speaking, not enclosed in a membrane. All right, so that's not a great diagram. It's kind of small to actually see that writing. I had trouble scaling that differently, but hopefully you kind of get the idea here. This gets ahead of ourselves a little bit because it talks about how it's anchored differently depending on what kind of cell wall the organism has. And I know we haven't got to that yet. We will shortly. But here's the filament, the hook region, and then this is just kind of showing how it's anchored into the cell wall. The basal body can be a little bit more complicated if it's anchored into a different kind of cell wall, the gram-negative cell wall. But they do show on this diagram here, this arrow, basically they're going to rotate. Okay, so it's going to move in a circular way, and that's how it propels the bacteria. As you see, that's different kind of motion than what we see for the eukaryotes. We have different terms as to how many flagella an organism has and where it's located. Okay, so if a cell just has one flagella, it's always going to be at one end or the other. It's not going to be, if it was sticking out like this, it would kind of move like a snowplow or something. So we don't ever see that. It's always going to be at one end or the other. If there's just one, we would call that a monotrichous arrangement. Okay, so mono meaning one, it's going to be polar. It's going to be at one end or the other. An amphitrichous flagella arrangement. Ampha means both. Okay, so one at both ends. That term amphibian, amphi, that means that's an organism that can live in both water and land. So prefix amphi means both. Lophotrichous, lopho means tuft. Okay, so a tuft of flagella at one end. And a couple people have asked me last semester, what do you mean by tuft? Maybe that's an old-fashioned word, but if you have like a tuft of hair sticking up, it's a clump. Okay, so it's a clump of flagella at one end. And then paratrichous flagella is a cell that has flagella all around the cell. Okay, so peri means around. The perimeter of the room is all the way around the room. Okay, so this has many flagella all around the cell. Okay, so we use those terms to describe an organism's flagella. And how they move does kind of make a difference as to how many they have and where they are, but we'll talk about that in a second. There's one other kind of flagella for bacteria, that the one that actually is enclosed in the membrane. We spoke about it briefly before. We call it an axial filament. Okay. And this is the kind of flagella that the spirochetes have. It's covered in a membrane. And we'll say it's actually composed of many endoflagella. Okay, so you can kind of see that in this diagram here. There's actually many little endoflagella in the axial filament itself, and all those little endoflagella apparently rotate. So that the flagella that you can see here is basically going all the way down the length of the cell. Okay, that's what propels that cell in a corkscrew-like way. Okay, so an example, I'm pretty sure That picture, yeah, that's the organism that causes syphilis, Treponema pallidum. Okay. All right, so we look at the motion of flagella. Basically, if an organism has one flagella and it moves in one direction, it goes one way. If it moves the other direction, it goes back the other way. So that's pretty simple. It's a little bit more complicated when it has a whole bunch of flagella. So cells that have paratrichous flagella, when they are moving all in a counterclockwise way, they all move together. Okay, and that we call that a run. Okay, when they're moving counterclockwise. Now, if the cell is sensing that it's not going towards an attractant, then instead of moving counterclockwise, the flagella will switch and turn clockwise, which basically all the flagella will do that kind of separately. So that will cause a tumble. Okay, so it will start out in another direction. It'll go in a run again. So it's a series of runs and tumbles till it senses that it's going towards the attractant. Okay, so that's what this picture is trying to show. In a run, the flagella are all coordinated, but see, the attractant is out here somewhere. So it needs to switch directions. It goes into a tumble. So again, a series of runs and tumbles. I think, let me show you, you can really get lost doing this for quite a while. I have a few, I can get it to come up, come on. Oh, there it goes. Okay. Let's see. This one's a little bit longer, but let's see. Okay, so that doesn't show up in the classroom very well. This one's a little bit darker. Here you can see, you know, they're buzzing all around. So if you watch really closely, often you can see it like tumbling around before it changes direction. Okay, if all those cells were just kind of floating one way, then those are cells that are not motile. That's when they're, again, moving. Sometimes they get stuck to the cover slip that's on there, and they kind of spin around for a long time. It's kind of fun just to see them buzzing around. Again, if you stain the cells, they're going to be dead. They're not going to be moving. So you have to either get your light just right on your bright field microscope, or use some dark field microscopy. There is another kind of microscopy, which I suspect that this is called phase contrast microscopy, that we didn't talk about. But okay, so that's kind of fun. We can watch that for a while. This, I think. The first one on there is just a little animation that you can watch sometime if you want. Okay, there it goes. I think this one, the cells were a little bit bigger. This is nice. It compares something that's oh, it's nice to put no background music. Oh yeah. So that's one that's probably sorry to get one. All right, I think you get the idea there. If you want to watch those later, you're welcome to do that. Okay, questions? Okay, so we'll move on. We'll talk more about that when we contrast it with eukaryotic flagella. Other structures that you sometimes see on the outside of bacteria cells, either fimbriae or pili. And these are two structures that sometimes we use those two names interchangeably. because they are both going to be composed of the same thing. They're made up of the protein pelin. Okay, so they have the same composition. When do we call it pili and when do we call it fimbriae? Well, usually fimbriae, we call them that if there's many of them all around the outside of the cell and they're fairly short. So in this picture, all these little yellow kind of hair-like appendages, those are fimbriae. These longer ones are flagella. Okay, so ignore those. Usually if these appendages are a little bit longer and there's only one or two, then we call them pili. Okay, so there's longer and there's only one or two. But these are not structures that are used for motility. The fimbriae typically are going to function in attachment. And it's very common, especially for gram-negative. There might be a few exceptions to that when I say that it's only gram-negative. And I know I haven't talked about the difference between gram-positive and gram-negative yet. We'll get into that. But very common, we all have E. coli in our large intestine right now that is staying in our large intestine because they have fimbriae to attach themselves to the wall of our large intestine. Okay, otherwise they would just be washed out down the digestive tract. Okay, so really important for cells to attach to whatever kind of cell or structure that they need to attach to. The pili have a little bit different function. Usually they're going to function in sharing DNA by a process we refer to as conjugation. Okay, so we're going to talk more about conjugation later on in the course, but it's a way that one cell can actually share some DNA with another. It will reach out its pilus, and then the pilus will bring those two cells really close together, and a pore will open up and it can exchange DNA. Okay, that's a process called conjugation. So anyway, these are two structures that we'll be seeing from time to time. All right, so that brings us to the cell wall. Yes, question. What did I mean by gram-negative? I'm going to talk about that right now. So I was getting ahead of myself a little bit. Yep, sorry about that. So the bacterial cell wall, this is a structure that we're going to spend a good deal of time on. It really, we have a lot of antibiotics that we use target bacterial cell walls. So it's an important structure to kind of break it down a little bit, what exactly is going on there, how it grows in the composition. There's really two major kinds of bacterial cell walls. I'm getting ahead of myself a little bit though. What's the function of the cell wall? It basically imparts the shape to a cell. So there are some cells that don't have a cell wall, and then those are the ones that we refer to as pleomorphic, but they really don't have any shape at all, or they could be many different shapes. Okay, so imparts the shape of the cell and imparts some strength to the cell and helps it withstand changes in solute concentration. We'll talk more about osmosis and whether a cell is taking in water or losing water. I'm going to review that either today or next time. But if the solute concentration is lower outside of the cell and water is rushing in, it's not a problem for bacterial cells because they have a cell wall. Okay, if that cell wall is damaged in some way, then that solute concentration can be a big problem. Okay. So no matter whether it's whichever one of those two basic cell wall types that we're going to get into in a minute, all bacteria that have a cell wall will have a layer of peptidoglycan. in their cell wall. Okay, and again, this is only bacterial domain. Okay, any archaea that have a cell wall don't have peptidoglycan in it. Okay, so this is something that's it's unique to bacteria. And if you look at this, the word itself, peptidoglycan. Let's break that apart. Glycan, when you see that root, that should make you think sugar. Okay, so there's actually two different kinds of sugar that makes up this compound. N-acetylmuramic acid. Usually we abbreviate that and call it NAM. And then there's N-acetylglucosamine. Usually we abbreviate that NAG. Okay, so these alternating sugars, they're actually sugars. I'm going to come back to the slide in a minute. But here's those two sugars. They're actually very similar in structure to glucose. Okay, so if you compare N-acetylglucosamine, it's only different from glucose at one of those carbons. Okay, and if you compare N-acetylmuramic acid, it's almost the same as N-acetylglucosamine, except for at this one spot there. Okay, so very similar to glucose. And then this peptido portion, that should make you think peptides or amino acids. Okay, the sugars are actually cross-linked by amino acid bridges. And we're not going to talk about which amino acids. There's some variation there. It could be different ones. So we're not going to worry about that. But amino acid cross bridges. Okay, so if you look at, here's a schematic of what we think peptidoglycan might look like. Again, alternating sugar subunits. These little balls here, those are the amino acid cross bridges. From one layer down to the next, and from one layer to the side to the side. So if you kind of think chain link fence, okay, it's a very rigid, very strong structure. Okay, it's permeable to anything that can fit through there. Okay, so the permeability, that's a cell membrane's job. The cell wall is pretty permeable as long as something's small enough to fit through. But it does impart a lot of strength and rigidity to that bacterial cell wall. I want to talk, your book, I don't believe brings this up. But think about a minute how that peptidoglycan grows. Because when one cell becomes two cells, that's called binary fission. That's how most bacteria cells reproduce. That one cell gets bigger and bigger, and then it splits into two. Okay, so a cell's cell wall has to be able to expand and get bigger. And that has to be done very carefully, because if there is any kind of little hole or any kind of fragile spot in the cell wall, then the cell is going to be much more susceptible to changes in osmotic pressure, and it can burst. Okay, so in order to grow, I want to look at a couple of the enzymes that have to kind of work in concert together in order for that cell, that peptidoglycan structure to expand. Autolysins, these are going to be enzymes that cut the bonds between the sugars. Okay, so here's a couple of the sugar subunits here. The autolysins are going to cut that bond. And they also cut the bonds between the amino acid cross bridges. Okay, so when that is done, it has to be done kind of carefully, not too much cutting of bonds at once. The transpeptidases, these are the enzymes that seal up, seal those amino acid bridges once again. Okay, and there's another enzyme that seals the sugars back together again that I just thought, well, let's not focus on that. Okay, this was, I'll come back to that in a minute. This was kind of the best picture that I could find to kind of try to visualize this, but here's inside of the cell. This is where those new subunits are made. So the NAGs and the NAMs are made inside of the cell. They have to be brought through the cell membrane in certain channels. That's BP actually stands for bactophenol, but you don't need to worry about that. But they're brought through certain channels. Cuts are made here in the cell wall, and maybe here's where a new sugar would be inserted. Another enzyme seals up those bonds. Here's TP, that stands for transpeptidase, that's sealing back up these amino acid cross bridges. Okay, so cuts, insertions, seals it up again. Okay, and again, that has to be kind of orchestrated really well. We're going to come back to this, but these transpeptidases, guess what binds transpeptidases? Antibiotics. There's a lot of antibiotics. All the beta-lactam antibiotics, like penicillin, bind to transpeptidases. And that makes it so the bacterial cell wall is not quite intact. Okay, so here, this is just a short, like 9-second animation of a cell bursting because its cell wall is not quite intact. Oh, come on. Oh, there it is. Okay. All right. So again, this is time-lapse photography. So the effect is not going to be quite as instantaneous as what it looks like. Bigger and then lysine. Okay, when they're in the presence of penicillin. Let me just show that again. Okay. That intact cell wall is just really important. Okay, there we go. Okay, so let's talk about the two basic kinds of cell walls. We have a gram-positive cell wall and a gram-negative. Let's start with the gram-positive. This has nothing to do with the charge of the cell wall. It's just positive for a gram reaction or negative for a gram reaction. Gram was the name of the guy that made up the stain to differentiate between these two different kinds of bacteria. So all bacteria cells have a slight negative charge to their cell wall. And remember, all bacteria cells have peptidoglycan in their cell walls. But they do differ as to how much peptidoglycan they have. Okay, with the gram-positive cell walls, they have a very thick layer of peptidoglycan. Okay, so the mnemonic that helps me remember that, plus gram-positive, that peptidoglycan layer is quite thick. Okay. What else is composed? What else is in there in a gram-positive cell wall? There's some sugars called teichoic acid and lipoteichoic acid. It's not really super clear what function they play. We think that these substances increase the rigidity of the cell wall. Okay, it may help in binding that cell to certain surfaces. It's not terribly clear. But gram-positive cells are going to stain purple when gram stained. And I'm not going to go through the steps of the gram stain. I did have that as one of my learning objectives. Don't worry about that. I'm going to leave that for your lab professor. I talked about that in lecture that was recorded. But basically, because the gram-positive cells, they have so much peptidoglycan in their cell wall, they take on the first dye that you add when you gram stain them, and they don't get decolorized very quickly at all. So they take on that primary dye, that purple dye. Okay, so here's a graphic of a gram-positive cell wall. Down here, this is the membrane, which we haven't gotten to yet. This whole part here, this is the cell wall. Here's some teichoic acid. And then the lipoteichoic acid means that it extends down and anchors into the cell membrane a little bit. That's really the only difference between those two. And of course, this big pink part, that's the peptidoglycan. Okay, so a major constitution of the gram-positive cell wall, it's mostly peptidoglycan. Okay, note that the shape of the cell, here they have it drawn as, hey, pretend this is a rod-shaped cell. Whether you have a gram-positive cell wall or gram-negative cell wall, that doesn't dictate the shape of the cell. Okay, there's gram-positive cells that are cockeyed, there's gram-positive, they're rods, there's other morphologies as well. Okay, the gram-negative cell wall gets a little bit more complicated. Okay. All right, so you have a thinner layer of peptidoglycan. Okay, so with that mnemonic, gram-negative, thin layer of peptidoglycan. And that's important because that means they don't hang on to that first stain. When you're staining them very well, they get very quickly decolorized, and they take on the last stain that you add when you're gram staining, what happens to be red. Okay, so they're red when they're gram stained. They have an outer membrane. So they have a plasma membrane and they have a layer of peptidoglycan, and then outside they have a whole nother phospholipid bilayer. Oops, I think that's bilayer. Okay. And that outer membrane has some foreign proteins that are basically transport channels. See a picture of those in a minute. And then on the very outside of the outer membrane, kind of extending in these kind of hair-like appendages, is a structure called lipopolysaccharide. Okay, we abbreviate that LPS, lipopolysaccharide. And lipopolysaccharide is composed of two polysaccharides and lipid A, which is also called endotoxin. Okay, so that's sort of a lot to throw at you. Let's look at a graphic of that and I'll talk a little bit more about it. So here's a gram-negative schematic of the gram-negative cell wall. Okay, working inside out, here's the plasma membrane or cell membrane, same thing. Okay, there's a little bit of what they call periplasmic space between the plasma membrane and the cell wall. Of course, this pink part that looks like a Wendy's hamburger patty, this is the peptidoglycan. And then outside of the peptidoglycan, this is what's known as the outer membrane. Okay, there's little purple globs in there. Those are some proteins. A porin protein looks like this. Okay, that again, that's primarily for transport through that outer membrane. And then these little kind of hair-like extensions, that's the lipopolysaccharide. Okay, right on the very outside of the cell. Let's take a closer look at that LPS. Okay. So on this slide from your textbook, again, this shows the LPS, lipopolysaccharide. So two saccharides. There's kind of a core sugar, and then this O antigen that's a second sugar. You may have heard there's an organism called E. coli O157 H7. Kind of a mouthful of a name for an organism. That is not the E. coli that you and I have in our large intestine. That's the kind of E. coli that you find in cows. When we ingest it by eating maybe hamburger meat that's not cooked all the way through, it makes us very, very sick. Like makes our kidneys shut down, like can be potentially deadly. That name of the strain O157, where does those numbers come from? Well, this is the O antigen. It's part of the lipopolysaccharide, and this organism has the 157th variation of that we found. H stands for different variations of flagellin. So it's the 7th variation of the flagellin protein that makes up bacteria flagella. So that in case you're interested, sort of thing. The real business part of the lipopolysaccharide, though, that we're going to come to back to lots and lots, is this lipid A. So that's the part that's anchored right in that phospholipid bilayer. And that lipid A is going to be disrupted or basically set free, let's say disrupted, whenever that cell wall is disrupted. So that happens mostly when the cell dies. Okay, if a cell dies, then basically the cell wall starts to break apart. Okay, and that's going to release that lipid A. I said that the other name for lipid A, it's also, we call it endotoxin. Okay, so remember, also called endotoxin. It's kind of a misnomer. Something called endotoxin you would think would come from within the cell, but when it was named, we didn't understand that it was actually on the outside of a bacterial cell. And say this was a gram-negative organism that was in our bodies, and say we took some antibiotics that had a massive kill-off of that gram-negative organism. Well, when their cell walls break down, they're going to release lots of endotoxin all at once. And that can be very bad for a patient. Endotoxin causes things like fever and inflammation. Okay, so if you have, a few cells dying here and there, it's not a big deal. It's a very low level thing. But if you have lots of cells dying all at once, Well, you have to take that into consideration when you're treating a patient with a gram-negative infection. The endotoxin causes a lot of inflammation. Okay, so again, lipid A is endotoxin. Okay, and we'll talk more and more about that. Okay, so here's the schematic of the two different kinds of cell walls that your OpenStax book shows you. Everybody draws things a little bit different, but here's again the gram-positive on this side, peptidoglycan. I got a whole bunch of it. They just drew the teichoic acid as little sticks and didn't bother to say the difference between teichoic and lipoteichoic. That's fine. And then the gram-negative, there's a few things here that we didn't really talk. We didn't talk about this murine lipoprotein, so don't worry about that. But we've got, again, a very thin layer of peptidoglycan, an outer membrane here, and it doesn't, well, it shows the lipopolysaccharide with the lipid A and the O antigen. So we've got all the parts there. There's actually a third kind of cell wall that's not nearly as common, but it seems like a good time to mention it. What we call an acid-fast cell wall. And I should mention that OpenStax calls cells with an acid-fast cell wall. It calls this kind of a subtype of gram-positives. Okay, so I'll just put that, some call these gram-positive because organisms with this kind of cell wall, they don't have an outer membrane. So, but they don't take up the gram-positive dyes very well either. So a lot of times we call this kind of a distinct, well, it's not really gram-positive or gram-negative, it's kind of its own special kind of cell wall. What's special about organisms with this kind of cell wall? Well, they have peptidoglycan in their cell wall, but they also have a ton of something called mycolic acid. And this is a long chain fatty acid that gives that cell envelope kind of a waxy quality. Okay, so you think about wrapping up a cell in wax paper, that's going to give it some little different kinds of qualities. Cells with this kind of cell wall are very resistant to lots of disinfectants. They are very resistant to drying out. They're just very environmentally tough. Okay, a cell that's coughed out, maybe it's an acid-fast cell and it's on some surface, it can stay viable for weeks, not just maybe a day or two, like if it had a capsule. Okay, so very environmentally tough. Maybe one drawback is that the cells grow really slowly. Infections caused by acid-fast cells tend to take a long time to kind of get established, but also it takes a long time to kill them. Okay? It takes a long time for the cells to manufacture those long chain fatty acids, that mycolic acid. And this is not common. There's really only one genus that's considered acid-fast, and that's cells in the genus Mycobacterium. Remember, underline whenever you're writing a genus name and capitalize it. There's a couple of really interesting infections that are caused by Mycobacterium. One is leprosy, and the big one is tuberculosis. Okay, so we'll be talking a lot more about TB later on in the semester. If you're in lab, you're going to get to do this special staining procedure to see these organisms. It's called an acid-fast stain. And we call it acid-fast because when we add a special decolorizer that's got acid in it, the cells have this acid-fast cell wall. They hold fast to the first stain that was on there instead of getting decolorized. Okay, so it's like they hold fast even when they are exposed to acid. And this actually is a really good acid fast stain. That was a picture that I took. The acid fast ones are bright pink, and the cells that are non-acid fast are blue. This was the picture that your book gave you. Not quite as good a picture, I think, of a stain there. This kind of cell wall is complicated. There's things here that I didn't really go into, but there is, again, this long chain mycolic acid, lots of peptidoglycan in there as well. All right, so if I kind of like this picture. It kind of compares all three of the cell walls, just all kind of drawn a little bit different. A few things on here that I didn't talk about, like this glycolipid, or again, some of these LAM. I didn't go into all that. But notice that, you know, the bright orange, that's peptidoglycan that's present in all kinds of cell walls. Okay, I want you to take a minute and just think about, we talked about these three different kinds of cell walls. What kind of organism do you think is going to be the hardest to treat with antibiotics, knowing a little bit about their structure? A gram-positive bacteria, gram-negative, or an acid fast. So I want you to just talk about that with the person next to you for like 30 seconds or a minute and see what you think. Okay, so let's hear some theories. Which kind of cell is going to be most resistant to antibiotics? What do you think? The acid fast. Okay. Why do you say that? Right, because I said it had a waxy coating and it was kind of resistant to disinfectant. That's true. That's true. Just the impermeableness of that waxy cell wall. And I tried not to say antibiotics when I was doing it before, but you don't have very many antibiotics that work against tuberculosis. It's a very narrow list, very difficult to treat. Now, between the gram-positive and the gram-negative, sometimes I catch people and they say, wow, they have all this peptidoglycan, it's very rigid, that must be hard. Yeah, but it's not impermeable at all. Okay, so between the gram-positive and the gram-negative, again, we have less options for those infections because of that outer membrane. And it's just a little bit more particular about what it allows through and what it doesn't. So again, gram-negative infections, a little bit more difficult to treat than gram-positives. Not that gram-positives are always easy. We have bacteria have a lot of other resistance mechanisms that we'll talk about. But in terms of cell wall structure, there we go. All right, good. Oh, and just to mention before we move into the cell, there is a genus of bacteria called Mycoplasma that does not have cell walls. Kind of sounds like mycobacteria, but different. Mycoplasma, that causes organisms in this genera can cause a walking pneumonia. You might have heard of that. Kind of hard to treat because a lot of our antibiotics target cell walls and they don't have cell walls. So they have these, you know, cells with all kinds of weird shapes and not really any set shape that we call pleomorphic cells. So just to kind of mention that those are out there. All right, so let's move into the membrane. I know we only have about 5 minutes left here. Not going to spend a lot of time here because bacterial cell membranes are very similar to our cell membranes. We have very few antibiotics that target membranes, again, because they're going to be toxic to us as well as bacteria because they're so similar. What does the cell membrane do? Its primary function is to be selectively permeable, to control what comes in and out of the cell. And also, as we'll see when we get into looking at metabolism, a lot of those metabolism functions of bacterial cells are going to be taking place in the cell membrane. Have to maintain concentration gradient. If there's a little hole in the cell membrane, then the cell is not going to be able to generate energy very efficiently. Okay. In terms of transport through the membrane, any type of transport that does not require energy is going to be considered passive. Okay, passive, no energy required. Oops. Okay, so obviously diffusion. We have a movement of some molecule that's in high concentration to low concentration. We call that down the concentration gradient, high to low. There are a few things that can diffuse right through a plasma membrane, things that are very small. Water can diffuse, although we call that osmosis. Things like glucose sometimes can diffuse, ions that are very small. Sometimes things that are lipid soluble can diffuse through the membrane. Oftentimes, though, some kind of protein channel is going to be necessary, and we call that some kind of facilitated diffusion. Okay, no energy. Again, when it's a passive process, no energy is required, but it just needs a particular channel. Maybe that channel is specific for one thing, or maybe lots of things can pass through that channel. And then osmosis is just the diffusion of water. Okay, let's take a few minutes to review osmosis. I always get students that don't have this straight. This is one of those concepts that you should understand as a science student, as a BMS student that's going through Grand Valley. I think where people get confused is when we use those terms, isotonic, hypotonic, hypertonic, those are describing the solution that the cell is in. Okay, and then if there's some solute in that solution, some solute, that means something that doesn't dissolve in the solution. Okay, so if you look at an isotonic solution, that's if the solute concentration in the solution is about the same as what's in the cell. Okay, no net change in where the water moves. If you have a hypertonic solution, and again, we use those terms to describe the solution because that's where the chemistry is likely to change really quickly. You don't say, oh, the cell is hypertonic. Okay, I mean, you could, but that's just going to confuse you. Okay, the cell chemistry is not likely to change. So use those relative terms to describe the solution. If it's hypertonic, that means the solution has lower solutes. Okay, so in this example here, sorry, I said it the wrong way. Higher solutes, higher solutes in solution. Just checking to see if you're paying attention still. Okay, so inside the cell is 20% solute, outside of the cell is 40%. So that means that the water concentration out is 60, the water concentration in is 80. So water concentration moving from high concentration to low, water is going to move out of the cell, okay, and this is going to dehydrate the cell. It's going to clean it, it's going to shrink in. That's very bad for a bacteria cell. Okay, they need water for their metabolism. In a hypotonic solution, lower solutes in the solution, and now just the opposite is going to happen. Water is going to be rushing into the cell. Now, if that was a cell without a cell wall, it wouldn't take very much water rushing in to burst the cell. But because bacteria have a cell wall, okay, they can withstand being in a hypotonic solution. Okay, they like a little bit of turgor pressure, just like a plant cell would. A little net influx of water is just fine, as long as their cell wall is intact. Okay, so there's another figure beyond this. It's in your book that just goes into that a little bit more detail that I didn't put up here. But okay, so again, look at that. If you make sure you got that square in your mind, I think there's a homework question about it. Okay, I think we made good progress. I'm not planning on posting any extra videos this weekend or anything. I think we'll get through it all on Monday fine. Start looking at the homework, and